Good afternoon, everyone. Welcome to the class of C3C1 Environmental Engineering Lab. Today we will perform the test for determination of turbidity. Perform the test to determine the turbidity in water. So we already discussed in our class that what is turbidity. Turbidity is mainly caused by what?
Suspend
Turbidity
of
is,
solid matter.
yes. So due to suspended solid, right? Suspended solid. That means if you have solid matter suspended in your water, then this turbidity. Okay, so it's a measure of degree to which water loses its transparency. If it's clear water, it's transparent, light can directly pass through. If this is turbid water, then light will scatter here and there. It will not directly pass through, right? So
Yes, sir.
transparency will lose. The transparency will be lost here. So it's a measure of the degree to which water loses its transparency due to the presence of suspended particles or particulates. Turbidity may be caused by a variety of suspended substances ranging in size from colloidal to coarse particles depending on the degree of turbulence. In river under flood condition, most of the turbidity will be due to relatively coarse particles, whereas in lakes and other waters, existing water relatively quiescent. Quiescent means calm. Okay, calm. So what they are telling here is this. You know, if this is a calm lake and it has turbidity, right? Now, if the water is not flowing, that means it's calm water or flowing at a very slow speed, then this suspended matter, due to their gravity, they will slowly settle down here, right? They will slowly settle down. And at a time, you will see that majority of the particles has settled down here. So turbidity is low. Turbidity is low here. Now, if there is turbulence in the water, for example, water is moving here and there due to high current. Okay, what would happen? Erratic movement of water. Then this particle, which were previously settled down here, particles which were previously settled down here, they will come again into the solution, right? So if it's a turbid water, I mean high turbidity, if there is high turbidity, no, I should say if there is high turbulence in water, if there is high turbulence, you know what is turbulence, right?
স্যার
Right?
টার্বুলেন্স মানে কি যখন হচ্ছে পানিটার স্ট্যাটিক অবস্থায় নাই তখন?
Yeah,
ওকে স্যার।
erratic movement here and there. Okay, high current. So higher is the turbulence, higher is the turbidity. Am I right?
ইয়েস স্যার।
Higher is the turbulence, higher is the turbidity, because the suspended material or particulates which are previously settled down now will come back into the solution. Now will come back into the solution and will increase the turbidity. That's what they are telling here. Environmental significance. Of course, always read the environmental significance of any test. Turbidity is important for water supply engineers as turbid water is not aesthetically acceptable to people. Of course, we will not accept turbid water. Also, for filtration process, turbid water is bad. So if you're using filter media, filter media, media to treat of turbidity, lots of particulate matter. Then eventually they will settle down here. They will settle down here and clog the filter very soon. That's a problem, right? Turbidity is also an important parameter in disinfection process. I discussed today in theory class disinfection process. What happens that if you have microorganisms in the water, bacteria, virus, then you use some chemical, for example, chlorine. So chlorine can kill these microorganisms. Are you following me?
Yes,
Yes,
sir.
sir.
Yes,
Chlorine
sir.
can chlorine can kill the my pathogens basically, and killing the pathogens is called disinfection process. Now, the thing is, if you have particulate matter, like solid, this solid, right? Yeah. Now, what happens that this bacteria hide, this bacteria hide in the pores of this particulate matter. Therefore, the chlorine cannot access them easily. Now, if chlorine cannot access them easily, this target group, then the disinfection will not be effective. That means the pathogens will not be killed. Therefore, if there is high turbidity in your water, that will reduce the efficiency of the disinfection process. Is it clear?
Uh,
Is it clear?
স্যার টার্বিডিটি বেশি থাকলে হচ্ছে ডিসইনফেকশনটা এফেক্টিভ হবে না, সেটাই তো?
Yes,
ওকে
yes,
স্যার।
yes. Because microorganisms are hiding inside the suspended particulate.
ইয়েস
Clear?
স্যার, আন্ডারস্টুড স্যার।
Okay, so disinfection is usually accompanied by means of chlorine, ozone, and chlorine dioxide. To be effective, there must be a contact between the agent and the organisms to be killed. However, in cases which turbidity is caused by municipal wastewater solids, the pathogens may be encaged. That's what they're calling encaged. That means hidden, okay, hidden in the particles and protected from the disinfectant. Disinfectant means the chlorine. In addition, the suspended particulate absorb heat from sunlight and making turbid water become warmer, so reducing the concentration of oxygen in the water. Now, what happens? Another problem of turbidity is that if you have high turbidity in water, if you have high turbidity in water, for example, river turbid, what happens? They absorb the sunlight. They absorb the sunlight, sunlight, and quickly heats the water. Quickly heats the water. Therefore, the temperature of water gets risen up by this suspended particulate matter easily. Now, a property of water is this: if temperature increases, then the dissolved oxygen carrying capacity of the water decreases. Now tell me, we need dissolved oxygen at least 4 to 5 for a healthy 4 to 5 for a healthy 5 to 6 like milligram per liter. Now, if it gets decreased due to high temperature, then it is a problem. The fish will die out. Are you following me?
yes, sir.
ইয়েস স্যার।
Okay. According to Bangladesh Environmental Conservation rules, drinking water standard for turbidity is 10 NTU. What is NTU? This is a unit. For example, the weight of mass is what? Kg. The weight of turbidity is in NTU. What is NTU? NTU is a nephelometric turbidity unit. Where does this name come from? Because we use nephelometric uh, method to calculate or estimate the turbidity in water usually, so it calls turbidity. What happens? That is very easy, very easy. This is your sample. This is the lamp or light source. If there is no turbidity, the light can directly pass through. But if there is turbidity, as you can see, black, so what happens? The suspended particulate matter reflects the light here and there, all through, all around, right? All around, all around. Previously, it was directly passing like this, but now, due to this obstruction offered by the turbidity, they will be scattered. Now you have a device here which can capture the light, detect the light. Now this distracted light will hit this cell, and you can determine if there is light scattered. Is it clear?
স্যার লাইটটা যদি 90 ডিগ্রিতে না ডিফ্রেক্ট হয় তখন কি এই গ্যালভানোমিটারটা রিডিং দিবে?
This is not about 90 degree of the scatter light. 90 degree is for this device is 90 degree. This device is 90 degree. Okay, the light doesn't need to be 90 degree. That can be here and there.
ওকে স্যার।
Okay, as long as the light hits this film, you will detect the
ওকে
light.
স্যার।
Clear?
ইয়েস স্যার।
So it's easy. I mean, it will be shown to you in the experiment in the lab that you have a turbidity meter. We use this all setup is in a turbidity meter. Okay, we call it turbidity meter. Call it turbidity meter. Okay. That's it. So another test will be performed today, that is total solid, dissolved solid, and suspended solid. These are very important. I just discussed today's theory class that by using total dissolved solid, that is TDS, this is an indication of what? TDS is an indication of what? Huh? TDS is an indication of what?
Salinity.
Salinity. Because salinity comes from the cations and anions, for example, sodium, potassium, calcium, Ca plus 2, magnesium, right? And also chloride, sulfate. So if you can measure them, these are basically dissolved solid. Remember, this is basically the dissolved solid. This is what? Dissolved solid. Now there are other types of solids. For example, this turbidity. The turbidity is not dissolved. It is suspended, right?
Yes, sir.
ইয়েস স্যার।
Am I right? So this is called suspended solid. Total suspended solid. We call it TSS. Clear? Now together, what is, if you add them, this TDS and TSS, TADS and TSS, this is what? Total solid, all kinds of solid. So this is total solid, TS. Is it clear, guys?
Yes, sir.
Yes, sir.
So Ts equals to Tds plus Tss. So you should go through these very well, okay? I already discussed these things. Solids are incorporated in water from its source, minerals with which has been in contact. So they can come from the minerals or from suspended particulate matter. Okay, now environmental significance. To determine whether the water is suitable for domestic purpose, it is required to know how much solid it contains, right? Because you see they have calcium, magnesium, and this can cause what? Hardness in water, right? We discussed it. Hardness in water,
Water.
right? Because they can interact or react with the soap and make them ineffective. So whether it is domestic, whether a water is suitable for domestic purpose, we need to know the TDS. According to Bangladesh Environmental Conservation Rule, potable means drinking water, should not contain more than 1000 milligram per liter of total dissolved solid. As I told you today, that if it goes beyond 1000, it will be really difficult or grueling to drink that water. Salinity, high saline water. Water with high dissolved solid generally is of inferior palatability, right? Palatable means it doesn't taste good and may induce unfavorable physiological reaction in the user. Concentration above 500 milligram per liter results in excessive scaling in water pipes. What happens that if TDS is very high, then during the passage of the water, this calcium magnesium may convert from aqueous form to solid form, like calcium carbonate or this thing. Now these are solid. So if this is solid, they are gonna deposit it. They're gonna be deposited here, and then your pipe will be, there will be a scaling. This is called scaling. Okay. So maybe acetic. Okay, so fine. So theory of experimental method. So how do we determine? We already know this, that TDS equal to specific conductance into 0.65. I already told you today, right? In that theory class, the TDS, total dissolved solid, can also be determined using electrical conductivity or specific conductance into 0.65. It varies. It may vary 0.55 to 0.90. This is very rare, zero point 0.9, maybe 0.55 to 0.7. Okay, so this varies. This varies. So into electrical conductivity. Now you have to remember that the unit of electrical conductivity or a specific conductor, same thing, should be in microsiemens per centimeter. So if this is in microsiemens per centimeter, then the unit of TDS will be in milligram per liter. Since this is empirical equation, you must use the designated unit here. Clear? So that's one thing. You can use an electrical device to calculate the electrical conductivity, and then from there you can calculate the TDS. Is it clear?
Yes, sir.
Yes, sir.
Now, the thing is, the second method that we will be using in the lab today is how to determine total solid, total dissolved solid, total suspended solid, and total solid. So if you know these two, you can always add them up and calculate total solid. That's not a problem. The thing is, we have to determine TDS and TSS. Okay, it's very easy. First of all, for example, this is the sample you want to measure the TDS for. You take a beaker, and that is basically 150 ml beaker. You take 100 ml. So you take 100. The capacity of the beaker is 150, and you take 100 ml of sample. From here, you take 100 ml. Okay. Now, before you take the sample, there is some issue. Before you take the sample, First of all, you put this in an oven. In oven. Okay? Dry it for 24 degrees Celsius. Sorry, for 24 hours in and around 100 degrees Celsius or 105 degrees Celsius. Dry this out so that there is no moisture. Okay. Now take the weight. Take weight of the beaker. That's W1. Is it clear?
Yes, sir.
And number 2. Now in this beaker you take sample from here. How much? 100 ml. Okay, 100 ml. Now if you have sodium, potassium here, and other dissolved solid. Sorry, you may have sodium, potassium. Also, you may have suspended particulate matter, right? You may have suspended particulate matter. So you have two things. You have T. Suspended particulate matter will give you what? TSS, and dissolved sodium, potassium will give you what?
TDS.
TTS. Fine. So you have both. Now let's put them in different color. For example, sodium potassium is red color and black one is the TSS. Okay. Now what you do, you put this in an oven again. Put it in oven. Keep it for 24 hours at 105 degrees Celsius temperature. What will happen? Tell me. From here, all the H2O will go out, evaporate, because this is 100 degrees Celsius beyond the boiling point of water. So H2O will go out. Am I right?
Yes,
Yes,
sir.
sir.
Fine. Is there any water now?
No, sir.
No. Now what is remaining here? Listen. When you put this in oven, H2O gets dried out. But this sodium, potassium, and this TSS, they don't go out. They don't go out. So what is remaining here? Tell me. The remaining here is the suspended TSS,
So total solids.
TSS, and exactly, and sodium, potassium. You have also sodium, potassium. Okay. So you have TDS also. So basically you have both. That means you have what? Total solid. You have total solid. Now, what do you have in total here? You have this beaker weight plus total solid. If you measure this now, what weight would you get? Tell me. You will get the beaker plus total solid weight. Let's call it W2. Clear?
Yes, sir.
Fine. Now, what is the beaker weight, guys? Guys, this is the beaker weight, right?
Yes, sir.
Now, what is the total solid? W2 minus W1 is what? W2 is beaker plus total solid, and W1 is only beaker. Then this is what?
Weight of total solids.
Weight of total solid. So this is the weight of total solid, maybe in gram or milligram, for example, gram. Now, if you want to find out the concentration of the total solid, what you need to do? What is the concentration? C equals to M by or weight by P.
MB. Sorry,
V.
sorry.
So weight is what? this W2 minus W1, and V is what? Yes, V is what? 100 ml, right?
Yes, sir.
This is 100. That's it. So this is the concentration of what? Total solid, right? Right?
Yes, sir.
Fine. So you know the equation. If you know any two, you can determine the other. You don't need to determine all the three directly into the left. You have this equation. If you know any one, you can determine the other one. If you know any two, then you can determine the other one, right? If you know these two, you can determine this. If you know these two, you can determine this. Am I right?
Yes, sir.
Okay. So what we do is, we know now we have determined total solid. So now we'll determine any one of them, this one or this one. This one is easier. Why? Let's see. TDS, to determine TDS. Now you do the exact same thing again. You do the exact same thing again, but first of all, you take oven dry the empty beaker and take the weight, fine, and then you take 100 ml sample. That's also okay. Now, before this third step, oven dry, you do another step here. Before doing 3, 1, 2, you have to do another step. That extra step is, now you need to filter this water. You have to filter them out. Okay, let's say it again. First of all, uh, first of all, you oven dry the empty beaker. You oven dry the empty beaker. Now, before going to this step number 2, you are what you have done. You have directly poured the sample here, right? In the previous case, you have directly poured the sample here. Am I right?
Yes, sir.
Instead of doing that, what you need to do is you need to apply a filter here. You need to apply a filter here and make it 100 ml. Now tell me, since this is filtered, all the TSS, suspended particulate matter, will now be captured here, right?
Yes, sir.
Yes,
So
sir.
do you have the TSS here in this water? No, you
No.
don't have the TSS here. The TSS is now only here. TSS. But TDS, these are dissolved, so they can easily pass through the filter. But the TSS, since they are larger, they will be captured in the filter, right?
Yes, sir.
Yes, sir.
Of course, this filter has a
Ji-
certain pore size, and that should be 0.6 micrometer. Usually 0.6 micrometer. So yeah, any type of filter will not work here. 0.6 micrometer. Okay. So now you have TDS. You don't have TSS here. So if you repeat the same procedure again, you will not have TSS here, only TDS. So you do the same. Now this will be what? TS or TDS? TDS.
TDS.
Now you do the same. This will be TDS. Do the same, it will be TDS. Is it clear? Is it
Yes,
clear?
sir.
Yes, sir.
Okay. So only the difference is nothing, but you do the same thing. 1, 2, 3. But before doing this, before pouring the sample, you just use a filter. That's it. There's no other difference. Clear?
Yes, sir.
Previously, When you determine Ts, you did not use the filter. But now when you're determining Tds, you use a filter. Okay. So now what you have determined? So far you have determined total solid and you have determined total dissolved solid. So now you know total solid equals to total dissolved solid plus total suspended solid. So if you know this and this, you just calculate this. Clear?
Yes, sir.
Yes, sir.
Subtract this from this, you'll get this. That's it. And then solid residual regard. Okay, if you want to previously we determined this in gram per liter, right? This is in gram and this is in milliliter. So if you want to have this in liter, gram per liter, so you convert this milliliter into liter, so you multiply it with 1000. That's how you get milligram per liter. Milligram per liter. So if you want to put the unit of solid residue milligram and the volume of the sample in milliliter, then you have to put 1000 to get it in milligram per liter. Okay. And
Okay, sir.
finally, Ts is equal to Ts minus Tds. That's it. Any questions?
No, sir.
Okay, so thank you very much. Have a good one and stay safe.